Okay, so hello everyone. My name is Yanis Asloudj. I'm a second year PhD student at the Labari and the BPH in France, so a computer science and health population laboratory in Bordeaux, in France. And my two PhD supervisors are Patricia Thébault and Flore Mougin. And today I'm presenting to you a work that is entitled Embrace the Bias, is the extrinsic variability informative in a single-cell RNA-seq clustering analysis. So I'm going straight to the point. Usually you do single-cell RNA-seq when you want to characterize a tissue at a cellular resolution. Basically, the way it works is that you sample cells within your tissue, and you will try to capture the transcriptome within each individual cell. And in the single-cell pipeline, you usually have a step that's called the clustering, where you will try to identify cells with similar molecular profiles, with similar transcriptomes, right? To do that clustering, you have multiple methods. Actually, you have more than 300 today in 2024. And here I show you an example on a single dataset of single-cell RNA-seq with 4 different methods. And each point is a cell, and the color corresponds to the cluster predicted for the cell, the group predicted for the cell. And you can see that the 4 methods have different predictions. So we have a lot of methods and they don't predict the same results. So that's an issue. And we will call here these differences in prediction the extrinsic variability. So usually in the state of the art, you have some ensemble algorithms that have been developed to try to integrate the predictions of multiple methods to generate a result that is robust to your choice of methods. But yeah, the way they work usually is that they try to minimize this variability, these differences in prediction. That is, they will use some sort of objective function or some sort of similarity matrix. And we have asked ourselves, well, instead of minimizing these differences, can we not use them? Can we not leverage them to enhance our analysis? And so we have developed 2 metrics and a new algorithm to try and leverage these differences. I'm going briefly over these metrics. You will see them, in the presentation of the algorithm. But basically, we have one metric called the overlap, where you will try to quantify the differences, or more so the similarity of 2 clusters predicted by 2 different methods. And because we don't just use 2 methods, but instead M methods, we have M clustering algorithm in our ensemble algorithm, we will have the consensus. And this one will quantify between 0 and 1 as well. This one will quantify the robustness, the agreement across the M method and the robustness of the cluster we predict with our own method. Okay, so keep that in mind. Overlap between 2 methods, consensus across all methods. It's the robustness of the prediction. So moving on, our algorithm, I'll skim over it. Feel free to contact me if you want to have more information. Basically, the way it works, you have your single cell data. You will cluster with 4, here we have 4, which will cluster with M clustering method to generate different predictions. And from these different predictions, you will identify consensus clusters. So you can see here on the top left, we have 4 methods and they predict different clusters. For instance, Sharp predicts the cluster A with cells 1 to 3, whereas Monocle also groups the cell C4 to the cluster of C1 to C3. And so the first thing we do is that we model the prediction we have generated as a graph, where each node is a cluster. We have cluster A here and cluster G. And we model the edges as the overlap, so the minimum proportion of cells that are shared between 2 clusters. And this allows us to have a graph with nodes and edges, and we can further filter edges that have a very low overlap, because sometimes you might have 2 clusters that are somehow sharing cells, but that might be due to randomness. And so we filter the low edges, and we only keep the strong edges. By doing so, with M methods, we will generate subgraphs. Here, for instance, in this toy example, we have two different subgraphs. By comparing the subgraph, we obtain experimentally to the optimal subgraph, that is, all the clustering methods we use, they predict the exact same clusters. In our example, with 4 different methods, the optimal subgraph would be 4 nodes with 6 edges, each equal to 1. And so by comparing the structure of our subgraph to the theoretical maximum, we can quantify the consensus between 0 and 1, and it corresponds to the robustness of our prediction. And so we look at the cells that are grouped together, we quantify the consensus, and we can further filter out low consensus clusters. And afterwards, all we need to do is to, well, try to characterize the clusters we have identified. We also generate a leftover cluster where we group all the cells that were not assigned to a cluster with enough consensus. And so we try to characterize them biologically. And if it works, we have multiple subclusters identified with an explicit robustness metric. And we can repeat this operation iteratively as long as the predictions increase the robustness. And so here, I'll try to go quick. We have applied it on a glioblastoma dataset, and the results were pretty good because we show that our method, when it predicts consensus clusters, well, they are homogeneous when you have one cell type. That's what you can see with the color scheme. Or here is the heat map. And we also show that we had 2 different subpopulations of cancer cells that were biologically distinct. One was more related to interaction with the immune system, and the other one was more invested in nervous biological process. And so we effectively address 2 main challenges with our method. That is, we explicitly quantify the trust we have in our results, and we deal with the varying levels of resolution by giving an objective argument to subclusters as a population of cells we identified. That's it for me. Here I show you this excellent article for single cell, and feel free to contact me if you want to further discuss this subject. Thank you very much.